Assalamualaikum warahmatullahi wabarakatuh and hi, we are from group 2. So in this video, we are going to present our lab assessment which is practical lab 4, digestions of starch and lipids. First of all, I'm going to introduce the group members. First is Auni Najiha binti Anwar. Second is Amir Khalid bin Baharudin. Third is Nur Widat Wafia binti Muhammad Amin. And last, me, myself, Nurul Shazliana Atika binti Abdul Hamid. So, in this video, we are going to explain objective of the experiments, methodology and result for experiment digestions of starch. Next, methodology and result for experiment digestions of lipid. And lastly, discussions for the experiments. The objective for the experiment is to examine the functions of a common enzyme in the digestions of starch and lipid. Second, to determine the factors of affecting
Assalamualaikum and hi everyone. My name is Auni Najah binti Anwar and you can call me Auni. My metric numbers is AS11540. So right now, I'm gonna show you guys our first experiment, which is experiment of digestion of starch. First and foremost, we're gonna start with experiment methodology. For this experiment, we have 3 steps, all of them. The first step is we need to prepare 5 pairs of test tube, and each of the test tube we need to label them with pairs A, pairs B, pairs C, pairs D, and pairs E. Why? Because for this experiment, we're gonna fill in each of the test tube with different kinds of solution. We don't want anyone to get confused, right? Okay, so for the first pairs, which is pairs A, we're gonna fill in with 4 ml of glucose, and second pairs B with 4 ml of starch, and the third pairs, which is pair C with 4 ml of untreated amylase, and the fourth is pairs D with 2 ml of starch and 2 ml of untreated amylase, and the fifth is pairs E with 2 ml of starch and 2 ml of acidified amylase. And after we have filled in all the solution in all of the test tube, we need to leave them for 10 minutes. Okay, after 10 minutes, we need to test each of the pair of tube with first pair with 0.5 ml of iodine solution, and next second pair with 0.5 ml of Benedict's reagent. And after we have tested all of the test tube with iodine solution and Benedict's reagent, we need to incubate in water bath at 100 degrees Celsius for 5 minutes. And after 5 minutes, we can see the result of the experiment, and then we need to record it in the table.
For the pair test tube A, there is no color change for iodine test, while for the Benedict test, the red brick solutions are formed. In conclusions, the iodine test shows absence of starch in solution, while the Benedict test shows the presence of glucose. For the pair test tube B, the solution turned to blue-black solution in iodine test, while there is no change for Benedict test. Iodine test shows presence of starch, while Benedict test shows the iodine test shows presence of starch, while Benedict test shows absence of glucose. For paired test tube C, there is no change for iodine test, while for Benedict test, the yellowish-orange solutions are formed. In conclusions, the iodine test shows the absence of starch, while Benedict test shows the presence of glucose. For paired test tube D, the blue-black solutions are formed in iodine test, while the darker orange solutions form in Benedict test. Iodine test shows the presence of starch, while Benedict test shows presence of glucose. For paired test tube E, the blue-black solutions are formed in iodine test, while the orange solutions are formed in Benedict test. Shows iodine test presence of starch, while Benedict test shows the presence of glucose.
Hi, Assalamualaikum. My name is Nurida Ofia binti Mama Amin. I will present to you experiment digestion of lipid. So my part is experiment methodology, digestion of lipid. The first step is 5 pairs of test tubes prepared as table 1. This is table 1. So the test tube are labeled A1, A2, B1, B2, C1, C2, D1, D2, E1 and E2. So the content for the test tube is for A1 in A2, 4 ml of glucose were added, and for B1 and B2, 4 ml of starch were added. As for C1 and C2, 4 ml of untreated amylase were added, and for D1 and D2, 2 ml of starch and 2 ml of untreated amylase were added. And lastly, for test tube E1 and E2, 2 ml of starch and 2 ml of acidified amylase plus 4 ml of amylase and 0.5 ml of 1 molar HCl. So for test tube E1 and E2, it have to mix well and leave it for 15 minutes. The next step is all the test tube must be shaken well and leave it for 10 minutes. The next step is the test reagent added as follows. First, 0.5 ml of iodine solution added into test tube A1, B1, C1, D1 and E1. The test tube must be shaken well and the observation must be recorded in the table. Next, 0.5 ml of Benedict's reagent must be added into test tube A2, B2, C2, D2 and E2. The test tubes also have to be shaken well and incubated in boiling water bath at 100 Celsius degree for 5 minutes, and the observation must be recorded in the table as well. Lastly, the table result must be completed for the observation.
So, the result for experiment 2, digestions of lipid by lipase. In test tube A, as you can see, the solution changed from brown solution to green solutions. And this is mean there is no reaction happen. For test tube B, you can see that the color of solutions become lighter. This is mean the reactions is happening. For the test tube C, as you can see, there is no change happen. This shows that the reactions does not occur. And lastly, for the test tube D, the solution changed from brown solutions two green solutions. This is mean there is no reactions happen.
Hi and assalamualaikum everyone. So today I want to discuss about the digestion of starch and the digestion of lipid for discussion. So before that, my name is Amir Khalid bin Baharudin and my metric number is AS11542. So we go to the digestion of starch for discussion. So as we know, the when we want to determine the presence of starch, as we know, the when the starch is present, the solution from brown color will turn to blue black solution. So the blue black solution form to determine the presence of starch, while the when you want to determine the presence of reducing sugar, the yellow color will turn to the orange color. Okay, so we proceed to the digestion of lipid for discussion. For test tube A, no reaction occur. As we know, for test tube A contain lipase and bile. For test tube B, reaction occurs but slowly. So for test tube C, no reaction occur because the enzyme lipase is absent in the test tube C, so the reaction doesn't occur. So for the test tube D, lastly, the reaction not occur, and because the optimum temperature for the enzyme to react effectively is 37. As we conduct the experiment, we conduct it in the room temperature, which is not suitable for the enzyme. So we do something wrong, or the examiners do the something wrong when the measured the solution not accurately for the test 2D. So that's an error mistake. So that's all for me. Thank you.